Imagine if you had the power to choose what makes you you. You could choose what color your eyes are, how tall you are, whether or not you develop genetic diseases, the list goes on and on. Well, you don't have to imagine anymore. In 2012, Jennifer Doudna and Emmanuel Charpentier introduced a new, more effective, and cheaper way to edit genes, called the CRISPR-Cas9 method. Their discovery is considered to be one of the most significant discovery in biology. Whether you support, protest, or have no clue what gene editing is, there is no denying that gene editing will and is changing everything. However, in order to explore the possibilities of gene editing and find new ways to reap its benefits, we have to understand what DNA and RNA is, and how CRISPR-Cas9 works. The same way all English literature is written using 26 letters, all human and almost all other animal DNA is composed of the same 4 letters: A, T, C, and G. Of course, these aren't some arbitrary letters. They are the 4 nitrogenous bases, meaning they contain nitrogen, adenine, A, thymine, T, cytosine, C, and guanine, G. These bases will always bond with their corresponding pair, or complementary base, making a base pair AT and CG. These base pairs are what make up the middle of the DNA double helix structure. So, how are 4 letters responsible for human life? Each group of 3 base pairs is called a codon. Codons are important because each codon codes for a specific amino acid. When multiple amino acids are linked together, it forms a polypeptide chain, which is a fancy way of saying a protein. Proteins make up the workforce of the cell. Different proteins have different functions, like forming cell structure, transporting nutrients, or relaying signals to other cells so they can interact. Of course, these base pairs aren't just randomly ordered. When base pairs are specifically ordered, and therefore code for a specific protein, this is called a gene. Our DNA is long, and when I say long, I mean that if you took all the DNA in just one human cell and stretch it out, it would be 2 meters long. A lot of that code doesn't do anything. Think of it this way. Let's say you have a cookbook that's 1,000 pages long. When you open the book, it all looks like gibberish. The letters don't make words, and nothing makes sense. But when you start looking through the book, you start to see instructions scattered in between all the nonsense. That's, in essence, what our DNA is like. There's a lot of stuff, but actual useful genetic code is nestled between a lot of stuff that doesn't really do anything. So, why keep all the non-coding DNA there? It's because our body likes having contingency plans, just in case our DNA mutates, whether it be from chemicals or radiation. Having all that extra DNA decreases the chance that the mutation will occur in a part of your DNA that codes amino acids, preventing things like cancers from developing. Though you have most likely had already heard of DNA before, there is a likely chance that you've never heard of RNA. RNA performs many roles, one of the most important of which is coding and decoding DNA. If the genetic code in our DNA is an instruction manual, then RNA is the molecule tasked with understanding what it says and telling the cell what it wants. If you look at RNA, you might notice that it looks like half of a strand of DNA. The structure of RNA is critical for one of its most critical functions, which will be explained shortly. Patience Young Padawan. RNA decodes our DNA and makes proteins in a 2-step process: transcription and translation. Remember how the middle of the DNA is made of base pairs. In transcription, the messenger RNA, mRNA, goes in between unbonded DNA base pairs and connect to their complementary bases to the DNA. Once the mRNA is detached, voila, we now have half of out base pairs from the DNA. Of course, having half the number of base pairs isn't a problem. The mRNA then travels to the ribosome. If you don't remember much from 8th grade science, this is where proteins get made, where it searches for the other half of the amino acid code in the ribosomal RNA, rRNA. When the complementary bases are attached, the transfer RNA, tRNA, then brings the corresponding amino acid to the ribosome. This process is called translation. This cycle of collecting amino acids and adding it onto the chain, called protein synthesis, continues until a protein is formed. As I said before, genes are parts of our DNA that code for a specific amino acid. But if you wanted to change what amino acids get coded, this is where CRISPR-Cas9 comes in. CRISPR-Cas9 is currently the most effective and efficient way to edit our genes. The method occurs naturally in bacteria as part of its immune system. Because of its ability to accurately locate target genes and cut them, Scientists continue to use this method to this day. CRISPR-Cas9 is naturally found in bacteria as part of its immune system. Every time a virus enters the bacteria inserts a copy of the invader's DNA into its own database, which is called CRISPR. This mechanism has a similar concept to vaccines. The DNA saves a copy of the virus's DNA. So the next time it becomes infected by it, it will be able to quickly identify it. Knowing what the virus DNA will look like is half the work. If the virus attacks again, the bacteria must immobilize it before it does harm. This is where Cas9 comes in. Cas9 is a protein with a specific function of cutting DNA, specifically the DNA of the invading virus. Inside the Cas9 protein is a guide RNA, gRNA, a special type of RNA that will have the same genetic code as the invading virus. Combined with the CRISPR database, Cas9 and its gRNA will be able to locate and cut the invading virus. If the same virus attacks the bacteria will create a new gRNA using transcription that has the same genetic code as the virus. The gRNA then goes inside the Cas9 protein and guides Cas9 to the virus, hence guide RNA. Remember that bases will only bond with their complementary pairs, so the gRNA will only bond with the target virus, similar to how mRNA will bond with the DNA during transcription. Once the gRNA has bonded onto the target virus, the Cas9 will precisely cut the DNA where the gRNA has bonded to. The gRNA and Cas9 protein then detach, leaving the cut virus DNA behind. The virus DNA then tries to desperately pair itself using a process called non-homologous pairing. The virus desperately tries to find bases to pair with the cut DNA, but this process is very error-prone, so the DNA is often rendered useless. Scientists have managed to harness the power of this natural process to edit our genes by changing the gRNA to match the target gene instead of the other way around. Because of Cas9's simplicity and precision, the cost of gene editing has lowered drastically from the previous method of hand-cutting the DNA. Instead of having viruses infect the bacteria, scientists inject the target DNA into the bacteria and allow CRISPR to make a copy of it. The next issue is making sure that non-homologous pairing doesn't occur. To combat this, scientists inject another piece of DNA with the desired sequence to ensure that the complementary pairs are bonded together. This means that scientists have the power to choose which parts of the genetic code can be cut, replaced with an alternative. Now that we've talked about what gene editing is, it's time to talk about why it's important. Here are some of the most impactful uses for gene editing. 1. Genetically modified agriculture without realizing. You may have eaten genetically modified agriculture. The power of genetic editing has allowed farmers to overcome modern agricultural issues. One of the biggest problems with agriculture today is drastic changes in climate because of climate change. Scientists have combated this issue by genetically editing different crops to survive in different climates. So agricultural areas that are heavily affected by climate change, whether it be drought, warming temperatures, etc., can still grow crops. 2. Alternative to vaccines. It is well known that getting your flu shot once a year is fairly successful in reducing your likelihood of getting the flu. Vaccines work by inducing the immune system to make antibodies to target a specific part of the virus that sticks out from the virus molecule, called antigens. However, in the cases of some viruses like HIV, vaccines are hard to create, as it is hard to target a specific antigen on an HIV molecule. Antibodies for HIV patients to control the viral load, meaning the amount of virus per millimeter in the bloodstream, already exists, but only lasts roughly 21 days. With the power of CRISPR-Cas9, Justin Taylor of Fred Hutchinson Cancer Research Center came up with the idea of genetically modifying these immune cells to automatically keep producing these antibodies, allowing for a one-time treatment of the disease. Though research is still being conducted, using gene editing to create vaccines for viruses that previously did not have any is game-changing. 3. Designer babies. Of course, when gene editing is mentioned, designer babies are most likely mentioned as well. The issues with designer babies is as much a practical debate as an ethical one. If we had the ability to determine what genes our children could have, of course we would want them to be healthy. Genetic diseases could be easily avoided pre-birth by genetically modifying a baby's DNA pre-birth. However, if you are already going to modify the baby's DNA, What's stopping you from making your baby physically stronger, more intelligent, taller, have blue eyes instead of green? This is the reason why gene editing is still subject to controversy among many. Although we may have power over these things, should we even edit human DNA to create an ideal baby? Are we robbing a child from their potential life? Or by not genetically editing a baby with a genetic disease, are we damning a child for life? Though genetic editing is becoming more common, we have a responsibility to determine how we use the power of genetic editing. The power to control our species' genetic future is awesome and terrifying. Deciding how to handle it may be the biggest challenge we have ever faced. Jennifer A. Doudna, no matter what your stance is with gene editing and genetically modified organisms, there is no doubt that gene editing is changing the world and the way we look at life. Our relationship with what we perceive as natural is changing, for better or for worse. No matter how exciting or terrifying, both really, our future is becoming with the power of gene editing. It's important that we are not ignorant of the effects that gene editing will have on us, but also the generations to come. Thanks for watching. See you on the next one. Oh, and don't forget to subscribe.